Right. Hello, everyone. Thank you for joining us today for the virtual demo of the OsmoTECH XT and the OsmoTECH PRO microosmometers. My name is Shweta Nair. I'm the Senior Product Manager here at Advanced Instruments for the OsmoTECH portfolio of osmometers that serve the biopharma market. And joining me today is Rick Zampa, our application specialist, and he will be performing the demonstration today for you on both of these instruments. So let's get started. So while we are going through this demo, just want to make sure you're able to address any questions that you may have during the demo. Please feel free to enter them in the chat, and we will pause at certain intervals throughout the session to answer them. There will also be some extra time at the very end for a Q&A. So please feel free to engage with the chat function on your display. Now, I want to take a few minutes to have a quick blinded poll question for you. So a poll question should now appear. And if you could take a few seconds to answer those questions, that would be helpful. So to kick us off on this demo, I'd like to first start with a brief introduction to Advanced Instruments as a whole. We're a 65-year-old global leader in osmolality testing, experts in cell line development technology, and liquid handling quality assurance. We support customers globally and have install base in over 90 countries throughout North America, EMEA, and Asia Pacific. We're really focused on developing technologies that help you streamline your workflow and ensure confidence in the results to help you and your work. Within our current offering, we have 4 different product lines that are utilized in the biopharmaceutical space. Today, our focus will be on the OsmoTECH osmometers. Oh, there we go. The OsmoTECH osmometer portfolio is the only osmometer portfolio that is specifically designed for customers in the biopharma market. And they are optimized for testing of biological samples, such as your cell culture media, your buffers, your aqueous drug products. And we also offer the Solentum instruments and reagents for the cell line development workflow, the Anoximat anaerobic jar system for bacterial cultivation, and the RTEL instruments for pipette and automated liquid handling calibration. All of these 4 product portfolios are supported by our very own dedicated Advanced Instruments trained field service engineers and have service contract options available to help you maintain the equipment and prevent any disruption to your workflow. Specifically for today, and especially for our OsmoTECH users in biopharma, we offer qualification and validation services to ensure regulatory compliance and help you get your instrument up and running quickly. Now, when we speak about the OsmoTECH osmometer portfolio, I'd like to spend some time discussing the application of osmolality testing for biological development and manufacturing of drugs. Now, starting from cell line development all the way through cell culture scale-up, osmolality can be used to confirm the concentration of the media, the feed, and can really help you gain insight into optimizing your scale-up step. In the downstream purification workflows, osmolality is used to really confirm your buffer concentration, help ensure your buffer exchange is efficient, and measure the concentration of your drug substance. And finally, in formulation, all the way from development, osmolality really helps you ensure you're formulating your drug correctly, ensure stability testing, and your final drug product. Now, osmolality is a critical quality attribute that provides valuable information about the identity, the safety, purity, and stability of your drug product. Therefore, for injectable drugs, the osmolality of the solution must be in a physiological range, or it must be close to the osmolality of your blood, since a higher or lower osmolality can cause pain at the injection site and create a hypertonic or a hypotonic environment. So as you can see throughout the bioprocessing workflow, whether it be upstream or downstream, osmolality has various use cases, and our instruments have been really designed to cater to the customer's sample types, unique regulatory environment, and data compliance requirements within each of these specific areas. Now, when it comes to selection of an osmometer for your lab, there are some features that you may consider, and these have been listed here in no specific order. Is the testing process simple? Is the device easy to use? Is the instrument accurate and reliable? How much volume would you have to dedicate for testing of osmolality compared to the other analytical parameters you may have in your workflow? How long does this test take before you're able to receive the data and analyze the information? How do we record the data that's generated? Do we record them electronically? Can it be integrated into my ecosystem for data management? Will this instrument fit into my workflow in terms of regulatory compliance perspective, meeting the pharmacopoeia guidelines? Those are all different critical conditions and considerations that you may have when selecting of an osmometer. And the OsmoTECH osmometer portfolio is really built to meet all of those considerations and address those concerns you may have as part of introducing an osmometer into your workflow. So as I mentioned, the OsmoTECH osmometers are really built for biopharmaceutical labs, keeping your needs in mind, and have been designed with a lot of the customer feedback we've gathered over the last few years. The testing process is fast and simple to really integrate into any workflow and throughput needs. They have flexible data connectivity options and a very strong and robust data integrity features. These features are adjustable to your lab-specific needs, so it's flexible as you may be in a non-regulated environment, such as an R&D discovery, or as you go towards GLP and GMP environments. Now, the OsmoTECH XT, you can see it tests samples up to 4,000 milliosmoles and improve testing performance with viscous and high concentration samples. That's one of the products that we'll be demoing for you today. All the OsmoTECH portfolio microosmometers currently support the different pharmacopoeias, such as your European, your US, Japanese, and Chinese pharmacopoeias. So what is the focus today? Today, we will see the OsmoTECH XT single sample osmometer and the OsmoTECH PRO multi-sample osmometer. To compare the instruments, starting with the OsmoTECH XT, this device is a manual single sample test. The OsmoTECH XT can test a very wide range of sample types from 0 to 4,000 milliosmol and has been specifically designed with the intelligent freezing technology to measure complex viscous sample types. The test time, as you can see, is anywhere between 150 seconds to about 190 seconds, depending on the range. It's a one-step direct testing method. So you have a sampler that you directly pipette your sample and introduce into the instrument for testing. It requires very small sample volume of just 20 microliters. And in comparison, the OsmoTECH PRO is a multi-sample. So you're able to test up to 20 samples on a turntable with automated cleaning between each of these samples. It's a quick 90-second test. And the range of operation for this device is from 0 to 2,000 milliosmol per kilogram. And the sample volume required, again, still very small, is 30 microliters per sample. Now, both of these instruments support the pharmacopoeia guidelines from US, Europe, Japan, and China, and additionally also ensure you're able to meet and support the 21 CFR Part 11 and EU Annex 11 requirements from a data integrity perspective. So with that, I want to make sure I have any questions answered. Do we have any questions, Rick?
No questions so far.
Perfect. So again, just a reminder, you have the chat function available for you to ask any questions during the demo. If you have any, we'll make sure we'll have them addressed as we go through. And with that, I'm going to hand over to Rick, who's going to start the demo. Thank you, Rick.
Thank you, Shweta, and thank you, everybody. I'm going to turn off my camera for now so I can switch to the correct camera. So hold on one second while I change my camera views here. Oh, didn't turn that off. There we go. All right. You should be seeing two instruments on the bench.
Yes,
at this
you
point.
see two instruments, and you have been spotlighted for everyone, so they should be able to see you bright and clear.
Great.
Thank you.
All right, thank you everybody again, and hello, I'm back in the picture. So we have the two instruments up here on the bench. As Shweta said, we're going to be talking about the OsmoTECH XT, which is the single sample instrument, and the OsmoTECH PRO, which is the multi-sample instrument. I have both the instruments set here on the bench. I'm going to start out with the OsmoTECH XT, the single sample instrument for this particular demonstration. Then we'll move to the OsmoTECH PRO and show you some of the differences and the ways different operation parameters between the two. So starting with the OsmoTECH XT, you can see we have the instrument here. You have your obvious touchscreen display on the top. Your consumables box sits up here in the top with your sample tips and your chamber cleaners. I'll go into that when I run a sample for you. Have a built-in barcode scanner. Hold on, let me do this. I just thought about this. There we go. That's better. So again, you have your sample tips and cleaners here in the box on the top. Touchscreen display, built-in barcode scanner. Here's your chamber, your chamber cooling chamber, and your slide to introduce your sample. You have your sample handler here, which is used to introduce the sample into the instrument. And that pretty much covers the overall look of the instrument for right now. Now, I have this instrument set up in its most basic format. So as Shweta was talking about, all these instruments are capable of complying with EU standards, FDA standards, pharmacopoeia standards, and those types of things. But the way I have this set up right now is it's in its most basic format. The reason I start here is to show you just how easy it is to use, say, if you're in a very early stage research and development level, and you don't need all of those compliancy features. The instrument setups, you're going to come up and run a test on it. I will go through and set it up so that you can see just how it will meet all of the requirements going forward. So you could have electronic signatures, usernames and passwords logged in, and different things like that, which would be more needed in the GMP environment. So to get started here, again, we have our instrument. You can see it says ready across the top. It's green. If I did want to log in, I could touch that little guy on the top there, and it would allow me to log in. I could also add a sample ID here by touching this when I'm ready to start a test. As we said, the instrument range is 0 to 4,000 milliosmoles. 0 to 4,000 milliosmoles. So as you can see, we have our range selector here, and it's in the low range. I can easily switch to the high range just by touching the button there. The low range is 1,000, or not 1,000, 0 to 1,500 milliosmoles. The high range is 1,500 to 4,000 milliosmoles. There's no need to calibrate between changing ranges. I can switch between ranges and run a sample, no problem. The instrument comes in fully calibrated and ready to run in either range from the factory. Again, I said this is sample ID. This is your menu button. We'll get into that in a minute. This is your consumables counter. This is to help with inventory control. Lets you know when your consumables are getting low in the box. That way you can make sure you have more consumables ready to go. So the first thing I'd like to do now is just run a quick test just to show you how easy it is to run a test on the instrument. So what I'm going to do is I'm going to take the sampler here. I have my sampler. I'm going to take one of the sample tips here, and I'm going to slide it onto the sampler, and it will snap into place. Sometimes you can hear it snap into place. You just want to make sure that it's all the way sealed around the bottom. I'm going to grab an ampule of my 290 reference solution here. To draw up the sample, I'm going to put the sampler in with the plunger down. I'm going to wet the sample tip to assure I get a good sample. I'm going to draw that out. Now, very important to understand here, I'm going to zoom in so you can see this a little better here, is you want to wipe off the outside of the sample tip. So you want to take a Kim wipe and just make sure you get any excess sample off the sample tip. Now, I don't see a drop on the top of this tip, but sometimes you'll have a drop on the top. And I recommend you just take the Kim wipe, don't put any pressure on it, and just drag it across the top like that. That way you just take off the drop that's on the top, but you don't wick out any sample, and that assures that you get your true 20 microliter sample. I'm going to zoom back out so you can see next. So I have my sample loaded, I'm ready to go. I'm going to remove my chamber cleaner from the cooling chamber. I'm going to put the sampler into the cradle like that. And to start the test, all I have to do is slide this up into the instrument. The sample does not get aspirated into the instrument. The test is being run in that sample tip that I put on that sampler. So what I'm going to do now, just because I want to, is I'm going to show you, I'm going to put in a sample ID. Once I start the test, I can do that. I use the built-in barcode scanner to scan the box of the 290 reference solution, and it's added the sample number 3MA029, which is the part number for that solution. So we'll zoom back in so you can take a look at the screen. You can see now it's gone to yellow here and says testing. We have our status bar or a progress bar here. It shows you we're at about 20% status. So for those of you who don't understand or may not know how freezing point depression works, what we're doing now is we're taking the sample and we're driving it down below its freezing point. So we're in this part of the little curve you see here. That's actually the freezing curve. So we're driving it down to a set temperature well below its freezing point. Once we get to that temperature, the bottom of this little curve right here, then we're going to initiate crystallization of the sample. We do that by knocking the sample. Hopefully you might be able to hear that when that occurs. When that knocking occurs, it releases its last bit of energy because it is still a liquid sample at that point. It is liquid because we want it to remain liquid so that we initiate the crystallization when we want it to crystallize. It then goes up to what we call the plateau. Once it reaches the plateau, we wait for that to stabilize. And once that's stabilized, we read out the result in milliosmoles per kilogram. I think you heard that knocking sound there, or hopefully you did. That means that we've crystallized the sample now. It's gone up to its freeze plateau. We're now waiting for that to stabilize. Once that stabilizes, we'll read out the result in milliosmoles per kilogram of water. So we're about 70% through. And we should be done in a second here. So like I said, the freezing point technology is the most reliable and repeatable method for checking osmolality. And as you can see, we got a 291. I was trying to get 290, well within the acceptable range of the instrument. We're trying to find plus or minus 2 typically. And so we're at 1 milliosmol outside of what our expected range is. So now, once we have completed our test, you can see this flashing. It says remove sampler and clean chamber. So what I need to do is remove this sampler from the chamber. Then I want to take one of these chamber cleaners, insert it into the instrument, and twist it each way a couple of times. Take it, flip it over, insert it, do the same thing, and leave it in there. Excuse me. The reason we leave it in there is twofold. One, it tells you when you walk up to the instrument that it has been cleaned. And two, it's a physical reminder to you when you come up the instrument, because you have to take it out, that you need to clean the instrument after every test. This is the one key maintenance point for the instrument. As long as you do this quickly after every test, like I just did here, the test ended within, less than a minute. I came over, I cleaned it, took care of it. You should never have any issues with the instrument. So now, the next thing we need to do is remove the sample tip from the sampler. So we have our, sorry for going in and out on you, but I want to make sure you can see this here. So now we have our sample tip. Now you can eject this by pushing down on this and it will eject it. I find that it's a lot easier just to take the top, give it a little push there, and it slides right off. Then what you need to do is wipe the little Teflon tip here, clean it off. Then you can put it back into the cradle and sit it there and it's ready for the next test. So that's how you run a test on the OsmoTECH XT. Like I said, in its most basic format, you just come up, load your sample, hit start, and away you go. Doesn't take much, very simple, very easy, and easy to clean and maintain. So now let's go into the menus and let's talk a little bit more about how we get this instrument, some of the features that will help you with the GMP compliance, your regulatory compliance, and other things like that, if you need to use those things. So I'm going to go into the menus. And first thing I'm going to do is go into settings. First thing we want to talk about is users. So we're going to go into the users menu here. I'm logging in as the administrator. Every time I log into a different menu, I will have to use my password. because that's all part of the audit trail and the GMP compliance and data integrity features to assure that that all changes in functionality have been logged and shows who, when, and where that was done. So you can see here we only have 2 users on the system, the administrator and the service user. If I wanted to add a user here on the instrument, I would put in a user ID, a password, and I would select what level of access they have. The instrument is set up to have 3 levels of access: operator, supervisor, and administrator. Administrator has access to everything on the instrument. Anybody set up as an administrator can make any changes to the instrument. Supervisors and operators, we can adjust what level of access they have and what menus they have access to, and I'll show you that in a few minutes. So I'm going to exit out of this. The other thing I want to show you here is you can also use LDAP or Active Directory to add users to your instrument. And what this is if you want to have your daily Windows login also be your login for the instrument, you can use that, and your IT people would set up your user groups and put people into the specific user groups, and then your user IDs and passwords are managed by your IT department in your facility and not on the instrument. So all of the passwords and password strength and everything and how often it gets changed is managed through your IT network versus on the instrument, which it could also be done on the instrument if you wanted to do it that way. Okay, so that is users. So users obviously very important. If you want to have everybody log in to run the test, you want to make sure that the right user's logged in running the test. So once we go from users, let's talk about, this is called the restrictions menu. And what this is how you basically set up the instrument to run testing. So again, I will log in here. Yeah, I didn't think I got that right. As you see, if I put my password in wrong, it doesn't let me in. You can try 5 times and then it locks the user out if they don't get the password correct five times in a row. So as I said here, the administrator is logged in as the administrator. I have access to all functionality of the instrument. So here's the user account. So you can see we have supervisor and operator. So I can choose what level of or what menus the supervisors and the operators have access to. This is all selectable by your administrator or whoever sets up your instruments. So I can choose to have the operators maybe only run testing and take everything off, or I can have them have the capability to only calibrate and run and test, or I can have the supervisors have access to whatever limited number of things I want, or everything if I wanted to. The other thing we have here is passwords. Again, if you're putting your passwords in on the instrument, this is where you would manage that process. You can set up your password strength along the side here, number of characters, how many characters, capitals, special characters. You can set up when the passwords expire if you choose to have them expire. And then you can have pre-expire passwords on creation. What that means is if I set up a new user on the instrument, I put in the password for them, they immediately have to change their password to on their first login. Next is our testing function. And as I said, I had this set up in its most basic format when I started the instrument up. So what I'm going to do now is I'm going to turn on the user ID and sample ID so you can see the difference in the instrument once we go back to the main menu. So now if I run a test on this instrument, it's going to require me to log in, and it's going to require me to put a sample ID in. Changing of the range, you can require a login for changing of the range, when I'm not going to turn that on because it just takes me more time to go through the demo. Auditing function. So this is where you can turn on your electronic signatures. Again, all of this functionality is selectable. You can turn it on, you can leave it, you can turn it off, you can have whatever you want based on your laboratory's needs. As I said at the beginning, I have it very basic to start with because maybe you don't need all the regulatory requirements. But however, if you're in a GMP environment, you're going to need most of this turned on to track logins, to track user IDs, to track date testing time, and then probably even require electronic signatures. So you can see you can either review the results or approve the results. You can do just one, you can do both. You can, as I said, leave them off if you want to. And then you can choose who has the capability to do those types of things. I'm not going to turn this on right now because I only have 2 users on the system, and I need to have multiple users to be able to set this up so that I can go in and show you how to review and approve results. But it's very simple. If I'm the one who runs the test, and I log in to say I want to review the test, it will not allow me. My username will not even come up in the menu if I'm the one who ran the test. You can allow comments on the results. So what that allows you to do is say something went wrong with the test. You put the wrong sample ID in when you were testing it. So you want to go in. You can hold on the results page, put in a comment, wrong sample ID, rerun test, or whatever you want to. Require reasons for all changes and settings. You'll see that in a second here when I try to log out and I hit save. And record all logins and logouts. So it's going to record every time somebody logs into the instrument or logs out of the instrument. So I'm going to hit save now because I changed that one thing there, and I'm going to put the reason for the change in now, and that's demo. And that'll be logged in the audit trail as well. So now we've looked at the restrictions menu. We have the instrument set up differently now. We're going to exit out of that. And you'll notice now that the bar across the top here is going to orange, and it says IDs required. So after restrictions, I want to go into the data management function here. Again, all of this is about managing the data for data integrity, data compliance, if you're in that environment and require that type of control. Now, the other thing to remember is even if you're not in that environment, if your instrument is going to, or your product is going to get moved on from phase to phase, it's good to start out with all of this capability in the instrument, because as it moves from phase to phase and requires more and more regulatory compliance, it's already built in the instrument. There's no need to change instruments along the way to get to the compliancy features you need. So it's a consideration you need to make early in the process to look at your overall process and how that's going to move through. So here's our data management menu. So you can see this is the data management. We're at the export button. What this allows you to do is export the results, events, users, or controls databases to a USB in either a PDF format or a CSV file. You can send it to a USB, or you can send it to a network folder. I'll show you where the network folders get set up. Everything can be sent to a network folder. One thing to understand about all of these databases too, as I'm talking about them, is they all have unlimited data storage capabilities. Excuse me. Thank you. So every one of these databases has unlimited storage available. So for the life of the instrument, it will never lose your data. And the other thing to point out here is, although we're exporting the file remains on the instrument. It never leaves the instrument. All the data on this instrument stays on the instrument. You can never delete it. You can't alter it. You can't change it or anything. It always stays on the instrument. And that's critically important, particularly if you were in an audit situation and they had some questions about your results. The FDA likes to see the original source of the data. The original source of the data is always the instrument, and it will always be on here because it has unlimited storage of data. So we have an export function. We also have a database backup function. The difference between the database backup function and the export function, the database backup function also backs up the configuration file of the instrument. So if there ever was a catastrophic failure, for instance, and you needed to reload the data back on the instrument, you would pull this backup file and you could reload it on the instrument. It has 2 ways of backing up. You have a scheduled backup or a synchronous backup. Again, it can be sent to a USB or a network folder. The scheduled backup, as it says, is once a day, once a week, or once a month. The synchronous is every 5 minutes to 60 minutes, depending on how you want to schedule that. Difference between the two: if you were to do it once a month, you would have 12 individual files, one for each month of the backup file, the newest being the latest backup file. With the synchronous file, you have one continuous file that gets rewritten every time it does a backup. It's really a preference of your organization whether you want individual backup files or you want one synchronous file. So the next thing is the import button. This is if you did have a catastrophic failure, you needed to reload. this is where you would load that database backup file back onto the instrument. Finally, just one last little thing here, single PDF result. What this does is it sends every result as a single PDF file to your network or your USB. It's almost like a electronic printout if you need something like that. We'll exit out of there. And finally, we'll go into the networking function here. And this is where you set up your network capabilities or your communication protocols for your instrument. So if you're connecting to a LAN network, this is where you set up that LAN connection, and that allows you to then set up the network folders for the backup, for the exports, and for everything else you're looking at. You also have an OPC-UA connection capability. This is a true bidirectional connection capability that we have on the instrument. This allows you to send information to the instrument, so you could stop the instrument or lock the instrument from a remote location if you wanted to. And it allows you that capabilities of true dual bidirectional communication. We also have a web server, and this allows you to just view the data, the results database on the instrument. It's a capability that you can be sitting at your desk and say, I want to see how the testing is going in the laboratory. I don't want to walk all the way over there. You can set up a web address here. You can log on and see exactly what's going on with the testing. Have an LIS function if you're working with a laboratory information system. You can easily connect to that and send your information to a laboratory information system. Okay, so one last thing we want to talk about is controls. Controls is where you would set up obviously the controls that you're going to run on the instrument on a daily basis or whenever you're running your testing. And the one nice thing about the controls, so you can see we have our controls set up. We have two controls set up in here. We have our 290 reference solution there, and we have a 500 milliosmol Cal standard set up. The nice thing about this is there's more than one nice thing. One nice thing is you can chart your controls on the instrument. So I picked our 290, and you can see how it's charted along here. We have this set up as 3 standard deviations. I can go down to 1 standard deviation, makes it a little bit easier to see the data. 1 standard deviation is plus or minus 2. So you can see the majority of data. We got one little spot there that's outside of our plus or minus 2, but otherwise everything is in our plus or minus 2 numbers there. And so you can chart your data and see if there's any trending going on. And like I said, you can go up to 3 standard deviations there and see where your data is at. So exit out of that. You can export that data. Oops, I think I hit that twice. Oh, no, I hit the... So this is good because this is what I wanted to go to next. What this allows you to do is set up an action item. Since this is a single sample instrument, the only thing you can do is issue a warning. So what you can do is if your controls are out of range, you can issue a warning, and that warning can mean whatever you want for the user. Maybe the warning says if you get an out of range result, you have to recalibrate the instrument. That's something that you're going to have to set up in the instrument. You can set that up as 1, 2, or 3 standard deviations. So in this particular case, if we were running a 290, we would always say to set it up as 2 standard deviations before you issued a warning, which was plus or minus 4 milliosmoles. And that would then issue the warning for you. We don't have anything turned on. I'm going to exit out of that. Edit, delete, chart. And that is your controls database. You can enter as many controls as you want. As I said earlier, unlimited data storage in this database as well. Okay, I'm going to move out. And what I want to do right now is I just mentioned calibration. So the instrument only needs to be calibrated once per year. And that is our recommendation. If you want to calibrate more, that's fine, but it should only require calibration once per year. And hold on while I type in. And the calibration function is very, very easy. It walks you through the calibration features. You can see here, we're in the low range, so we're going to use a 0, a 500, and a 1500 milliosmol calibrator. If you want to calibrate more frequently, as I was saying just a few minutes ago, that's fine. We don't recommend calibrating more than quarterly. It just does not require it. The instrument is robust. It doesn't need to be calibrated frequently, And truthfully, it really only needs to be calibrated once a year. I can't even remember the last time we calibrated this unit, and you can see I get 291 on a 290 standard, and so it's very, very robust instrument. So say I started this calibration and I got halfway through it and somebody called me, I was late for a meeting and I had to leave it. I can easily just cancel out of this calibration. Yes, I want to cancel. Nothing has changed in the instrument. The instrument does not change the calibration curve until you complete the calibration. So if I wanted to continue to run this instrument with the old calibration, I could continue to run it. Nothing's been changed. It hasn't started changing any calibration. It will not make any changes until it says calibration complete. So the instrument is still functional, even though I canceled out of the calibration. Okay. Results database. This is where obviously all our results go. As you can see here, I ran the 3MA029 as we started our demonstration. It shows you the date, the time. No user was logged in because we didn't require it, but it shows you the lot number because I scanned the barcode in. You can also run statistics from this menu. So if I can hit it here. Oh. There we go. So I've picked 3 results. I can hit the statistics button and it automatically displays the statistics for me. In this particular case, we're really, really good. We got a 291, our standard deviation, and our CVR 0. So it's repeating really, really well. I could export this if I wanted to, if I had a USB or network folder for it, or I'm just going to exit out of it. The results database looks similar to an Excel spreadsheet. You can sort by any one of these columns. You can search by lot number, user ID, sample ID, whatever you want by using the search button. And again, stores unlimited test results. If you had controls in there, so if this is a control, I could chart my controls from here, or I could export any part of the database that I wanted to from here as well. Okay, one last thing to show you here is the events database, which is our audit trail. The audit trail is, again, unlimited data storage, and again, looks similar to an Excel spreadsheet, and you can do search by type, you can search by date and time, the user ID, anything in these columns, you can, by the descriptions, you can search and try and find whatever you're looking for in the audit trail. So you can see the logins and logouts, log into settings and controls, log into settings and controls. And down here, you can see where I changed user ID requirement from off to on. So it tracks every change in the instrument, which is required when you're looking at an audit trail from a regulatory standpoint. All right, exit out of there. So go back to the, I know what I wanted to talk about just briefly so that it's a little bit easier to understand. I'm going to go into settings. So you can see here on the settings database, or on the settings menu, we have these different key colors here. The red keys means only the administrator can get into it. The gold key means the administrator who always has access and the supervisor have access to that. And 2 keys, gold and silver, means supervisors and operators have access to that menu as well. So it just gives you a quick view of who has access to what in there. So now you can see the instrument is set up to require user IDs and sample IDs. And if we were to run a test right now, I would have to log in and do that. So are there any questions on the OsmoTECH XT before I jump over to the OsmoTECH Pro and just show some brief differences between the 2 instruments?
Yeah, perfect. So we have a question that says, are there any limitations on the number of users that can be on an OsmoTECH XT?
No, there is no limitation on the number of users. The database will store as many as you want.
Perfect. And you mentioned the XT is able to do challenging materials. What about semi-solid, like gel-like materials?
Very good question. Yes, it will handle gel-like materials. Actually, I just recently did a demo for a customer, and we had a solution that was in a tube, and when you turned the tube sideways, it didn't move. So yes, it will do gels. It does gels quite well. As long as you can pipette it basically into this sampler, if it will draw it up into the sample, we are pretty confident you'll be able to test it.
Are there any specific precautions that you require when you're testing such samples, Rick?
I'm sorry, say that again?
Are there any specific precautions to take when you're testing such samples or anything to be aware of?
Precautions, not necessarily. I mean, you want to make sure that the sample is at room temperature, if that's what you're talking about, because you want to ensure that you're getting a good freeze curve. If the temperature of the sample is too cold, it will give you freeze errors, because if it doesn't see a change in temperature quickly enough, it's going to assume that the sample has already frozen. So I guess that's one precaution you wanted to make, is make sure samples are at room temperature.
The other from my side would be just to make sure you're cleaning really well between the samples. So definitely, as Rick showed you with the chamber cleaner that's over there, you can dip one end of the chamber cleaner in purified water and do a wet clean, followed by a dry clean by reversing the other end of the chamber cleaner. So making sure the cleaning is done really well between testing of such viscous or gel-like samples would be critical to ensuring precision.
Okay, yep, I agree with that on that, Shweta. Sorry, I didn't quite understand where we were going with that question. So
that's all right. Another question is, can we test or analyze cell culture media after filtration? And yes.
Yes,
we can. And
that's one of the biggest applications for the osmometers, is cell culture media.
Correct. And another question was, do we also can we also test cell suspensions, and if there's any limitation to cell concentrations? There is no specific limit as such
Correct.
that we define for cell concentration, but we do have customers who do test with cells in the sample as well as cell suspension. Yeah,
we have a number of different customers who do different types of suspensions as well, as not just cell suspensions, but other types of suspensions as well, and we're able to test those.
Do we provide an IQ/OQ document for instruments?
Yes, we do. We provide a comprehensive IQ/OQ document, and whether it's one of our direct service engineers or one of our distributor service engineers, they'd be the ones that would come in and take care of that IQ/OQ for you and get the instrument ready and up and running for you.
Perfect. One more question: how frequent should we be using the standards and controls, and what are the recommended concentrations of these standards?
Okay, so standards and controls should be used every time you run the instrument. If you're trying to comply with USP, EP, or one of those pharmacopoeia standards, each one has a slightly different specification on how the controls are supposed to be used. But generally speaking, you'd want to bracket your sample value with controls. So if you're in this particular case, let's say our sample is 290, you'd want to do something above that. So say maybe a 300 or a 400, and something below that, maybe a say 200. Some of the references say you should be, no, they changed the 50. It's wider than that now. So yeah, if you're within 100 milliosmoles of each one, you're good in bracketing your samples. We also recommend that you run the 290 every time you run the instrument as an instrument check, to assure that your instrument is operating properly, because that's how we recommend that being done. Did I cover all of that? I think so.
Yes, I think you did.
Okay.
Perfect. I think that's good for now. If you want to jump over to Tech Pro.
Okay, let's move over to the Tech Pro. And there we go. So this is the OsmoTECH Pro. And again, we're going to zoom in a little bit here first so we can get started. So again, touchscreen display. You can see it's very similar to the OsmoTECH XT. The one big difference is we have the turntable here. The instrument, you can load up to 20 samples on the instrument at one time and run those 20 samples. You have each one of these buttons. You can look at the test result by clicking on the little button. You also have the positions over here that have been tested. You can see that all the positions that have been used, so it always goes to the next open position for you. You have your counter of your consumables again. So the consumables on this is a box of 500 sample cups and a cleaning ring, or a wiper ring as we call it. you can, when I zoom in, you'll be able to hopefully see. So the wiper ring is here on the turntable. You might be able to see the white spot there. That's the wiper ring. After every sample, the sample probe punches through this wiper ring and cleans the sample off and assures that you don't have any carryover. Very similar to what I did with the chamber cleaner, only this is automated and on the turntable here for you. So you can see the instrument says it's ready to go. So what I'm going to do is I'm going to run one test right now, because this instrument I have set up to require user IDs and sample IDs. So you can see the slight difference in the way that works with the instrument. So I'm going to, excuse me while I step in front and put my pipette tips on the wrong side. I'm going to grab my 30 microliter pipette. I'm going to grab a sample cup. All right, I'm going to pipette my 30 microliters into the bottom of my sample cup. All right, I have my 30 microliters. I'm going to hit my start button here. And you can see it brings up a screen, asks me to log in. So I'm going to log in as the administrator. Hit enter. It's going to move the turntable around to the next open position, which was position 8. I'm going to drop my sample in. You see it brings up sample ID. I'm going to take my, there we go, scan in my sample ID, and now I'm ready to run the test. I'm going to hit the go button, and now it's going to start running the test. So like the previous instrument, we're now doing, so like the XT, we're now going through our freeze curve. We're going to start cooling the sample down. You can see our progress bar along here. And the other thing I want to point out now is if I had multiple tests already lined up in the instrument, what this instrument allows me to do is add more samples to the instrument. So say I had 8 samples already loaded in there, and I came back to the instrument and I wanted to load more samples. I would hit this plus button, and I'm going to hit it now. And what it would do is it says wait. It's going to run the test that I'm on. Then it's going to finish that test. Then it's going to come back, and it's going to come to the next open position and allow me to add more tests. What this allows you to do is, say somebody loaded 8 tests on and you came in and you wanted to run yours, you don't have to wait for those 8 tests to finish to add your samples. You hit that button, it comes in, maybe you add 5 more, you hit start again, and it goes back to where it left off and finishes the testing. In this particular case, obviously I've only got 1 sample in, but it's going to still ask me if I want to load another sample here at this time. So you can see we're at about 70, almost 70% here. We should hear the knocking sound pretty soon on this instant. There we go. Now it's seeking it's looking at the plateau, measuring the plateau, waiting for that to level off. And then we should get our sample, our test result here, and we get a 290. So we got 291 over here, 290, slightly different instruments, slightly different sample volumes, but 1 milliosmo really is very, very good comparison. So you can see now it's cleaning between the sample. It's very hard to see that, but in the back of the instrument, it's going through, it's cleaning. So you see, because I came up to it and told it I needed to load more samples, it's now asking me to log in again, because it doesn't know who came up and said they wanted to load more samples. So it does not assume that it's the same person who loaded the first sample. So it asks me to load more samples. I'm just going to exit out of this because I'm not going to add another sample. But then it would also then walk me through the sample IDs, and then it would start the tests again. So this instrument has basically the same menu structure that I just showed you on the OsmoTECH XT. I'm not going to go through them all again, but I'm just going to show you quickly that the menu are pretty much the same setup. Again, calibration once per year on this instrument as well. You have your results database. Same configuration, except on this particular one. Come on. So on this, the only difference here is you see you have a position column here, which is the position on the turntable. You still have your user ID, your description, your sample ID, everything else in there is the same. If we go to our settings, you'll see it's the settings buttons are basically the same. You have users, controls, restrictions, networking, data management, everything that we talked about on the other instrument, all the same features and functionality there, with electronic signatures, user IDs, sample IDs, as I showed you on this particular one. And then finally, diagnostics, you have the same setup. You have your events database there, and it would show you the same types of things on the events database as we showed on the OsmoTECH XT. Again, major difference between the two is obviously one's a multi-sample and one is a single sample instrument. The capability to load 20 samples at one time and walk away, and then have it download the data to whatever network or USB spot you want it to load to. So with that, I will ask for questions about the OsmoTECH Pro. Again, functionality and features between the two instruments from a software standpoint are identical. So everything I showed you on the OsmoTECH XT applies to the OsmoTECH Pro. So with that, we'll go to questions.
All right, we currently don't have any questions.
No questions.
But feel free, if any of you have any questions, feel free to enter them in the chat, and we'll take a few minutes to answer that. We do have another poll question, so I'm going to have that poll question pop up while we wait for some of the other questions to come in. So if you could take a few minutes to answer the poll question, a few seconds actually, not a minute.
Yeah, hopefully it doesn't take you minutes, right?
Yeah.
Yeah, so both, like I said, both of these instruments have the identical features and functionality from compliance, data management, data integrity standpoint. Again, the great thing about the instruments is the flexibility which you have to set them up. You can set them up as easy to use as somebody just walks up and loads in a sample, or I don't want to say, or to meet as much compliance as you need with user IDs, sample IDs, electronic signatures. And it's important to understand that once you start looking at instrumentation in your processes, that this functionality is going to be required at some point in your process. And having it early in the process is key to making the transition from one phase to the next easier, because then they don't have to make any changes to any instrumentation being used.
Oh, perfect. Rick, we have a question. I think this person's probably testing some samples in the clinical environment, but just if they have an Osmo One and they're running samples, patient samples, and taking them in duplicates and taking an average, is that necessary with a multi-sample osmometer?
Is it necessary with a multi-sample osmometer? If you're running duplicates, yes, you should always run duplicates. And well, this is the OsmoTECH Pro. It's not used in the, but if you were to use the multi-sample instrument for the clinical environment, you would load 2 samples and you would run them in duplicate back to back, just like you would do on a single sample instrument. And that's another thing to point out. That's actually something we didn't talk about when we were talking about compliance. Is many of the compliance organizations and the pharmacopoeias recommend that you run everything in triplicate. And so you have to keep that into consideration when looking at the osmometer you're selecting. Is you may have only a few samples to run, and you might feel that the single sample is best, but with those few samples, if you're running everything in triplicate and running your controls, maybe a multi-sample is the better option, so that you can just load everything on at one time and let it run, and then come back and get your results when that's finished up. So if you had 3 samples, you were running those in triplicate, that's 9. You're running your controls, 2 controls in triplicate. Now you're up to almost 15 samples, or 15 samples that you need to run just to run your 3 samples and meet the pharmacopoeia compliance.
Perfect, thank you. And just a quick note for customers, those of you who have maybe more samples that need to be tested and are still doing triplicate, we do have a demo coming up for a high throughput instrument, the OsmoTECH XT, which does let you not have to individually load the replicates, but just load 1 sample, and the instrument will hold the replicate samples as needed for testing. And that's on October 8th, I believe. So please do keep it 9th. Sorry,
October
keep
9th.
an eye out for that. But yes, let me just see any other questions. No, I don't think we have any other questions. But again, thank you so much for taking the time and joining us today for the demo for the OsmoTECH XT and the OsmoTECH Pro. Thank you, Rick, for doing the demonstration. And to all of you who may not have questions now, but may have questions in the future, please feel free to reach out to us at aicompanies.com. And we look forward to seeing you soon. And stay in touch.
Thank you, everybody.
Thank you.
Have
Bye.
a great day.